Hello, good evening all. Am I audible? Can anyone confirm whether I'm audible or not? Hello. Okay. So I'll start sharing my screen. Is my screen visible? Are you guys able to see my slides? Please confirm whether you are able to see the slides or not, so we can proceed with our today's session. Are you guys able to see the lecture slide?
Yes, ma'am.
Okay. Thank you. Just I hope I'm still audible. Okay. Good evening, all. So, again, I welcome you all in today's problem solving session for week 2. And the lectures we will be covering for this week is lecture 1 is breakage of genomic DNA, and the lecture 2 involves repair of genomic DNA. So, this course, the genome editing and engineering, is being taken by Professor Utpal Bora. He is a professor at Department of Biosciences and Bioengineering at IIT Guwahati. I'll briefly introduce myself as well. I am Pooja Yadav. I am a PMRF fellow and a PhD scholar in Department of Biosciences and Bioengineering at IIT Bombay, and I am a PhD student in molecular virology lab. Like previous session, again we will be having questions. Again, I first like you to try to answer the question, and then we will look into the concept for each of the question. But first, please try to answer from your side so that we can have an interactive session. So let's begin with our today's session. So you guys are still able to see my lecture slides, or they are not visible? I think screen sharing is please interrupt me if the screen sharing is stopped, because I don't know why it is happening. It is happening from past few days. So please let me know if you cannot see my lecture slide. Please interrupt and just let me know. I hope I am audible. I Can anyone please confirm whether I am audible or not? Okay. Thank you. Let's begin with our session. Uh, so the first question for today's lecture is which one of these is not an endogenous source of DNA damage? First option is DNA base mismatch, B is DNA methylation, C is ultraviolet UV radiation, D is topoisomerase DNA complexes. Please try to answer the question. Yeah. Okay, so we have one option, UV radiation. So is there any other option that might be correct apart from option C? Okay, so the correct option for this question is option C, UV radiation. So apart from UV radiation, the other three are endogenous source of DNA damage, but the UV radiation is exogenous source of DNA damage. So we will look into what is DNA damage and what are these sources of the DNA damage and what happens when our DNA gets damaged. So we know that DNA is a reactive molecule. So both endogenous, including replication error, DNA base mismatches, topoisomerase DNA complex, deamination or the methylation of a DNA. These are the endogenous agent that leads to the breakage of our DNA, while there are other agents which are exogenous, which includes the UV radiation, uh, ionizing radiation, alkylating agents, etc., which damages our DNA. So, what is actually a DNA damage? It is the disruption or alteration of a basic chemical or the physical property of a DNA that can affect the normal function of a gene. So, by these endogenous or the exogenous factor, our DNA get damaged. It uh, can either have a single base pair mismatch, or it has a single strand break. So we know that in human we have double stranded DNA, so it can either be single strand break or it can be double strand break, or the two bases are cross linked together, or there is the formation of the adduct. So these are the common DNA damages by the exogenous as well as the endogenous factors. So, uh, so endogenous factors can be replication error. There is a possibility where a DNA polymerase has become less. Just wait a second. Somebody wants to join. Okay, someone has it. Okay, the screen sharing has been stopped. That's an issue. Oh, please stay here. I will try to join via different browser. Just wait for a while. Maybe I can solve this issue. I hope my slides are visible now. Sorry about that. So we will continue with our session. So there are certain DNA damaging factor which can be endogenous, which might present inside our cell, and there are other factors which are exogenous, which are present in our environment. So the endogenous includes the replication error, where there is a wrong base incorporated while the replication due to some reason, or there might be or there are exogenous factors which are ionizing radiation or a UV radiation which can break our DNA strand. So these are the DNA damaging agents, and which results either in base mismatch. The most of the endogenous factors leads to the base mismatch of where there is a different base is being added in place of some other base, or there can be a single stranded break or a double stranded break of a DNA because of some exogenous DNA damaging factors like ionizing radiation, or the bases might be cross linked, so there will be further issue in replication or transcription process, and or there might be the formation of adducts which further lead to hindrance in the replication and the transcription of the DNA. So the endogenous sources of a DNA, they originate within the cell. They usually result of a abnormal metabolic processes, and the exogenous sources, they came from the external environment factor. So the first option for this question was DNA base mismatch. So this DNA base mismatch occurred via the endogenous factor. It occurs during the DNA replication when a DNA polymerase incorporate different or incorrect nucleotide or a base pair. So let's suppose we during the replication process there is C present, and during the replication the complementary nucleotide would have been G, but if the error has occurred, there could be a different incorporation of a different base pair, let's say A in place of G. So this is a mismatch, a DNA base mismatch, because this instead of G, the A has been incorporated because of the error by the DNA polymerase. So this is the DNA base mismatch. It is occurring from the cellular source only, not coming from an outside. It is an endogenous factor. The second option was DNA methylation. It is again endogenous. Naturally occurs as a part of gene regulation, but abnormal methylation can lead to DNA damage or gene silencing. We know that so DNA methylation. DNA methylation occurs at we know that during transcription or the regulation of transcription, the RNA or a DNA, they tends to get methylated. They are a part of gene regulation, but the abnormal methylation of a DNA, it can be damaging for the DNA. It can lead to the gene silencing as well. So this DNA methylation is also endogenous, not exogenous. The third option was ultraviolet radiation. It is exogenous. It comes naturally from sunlight. So it causes the thymine dimer formation and other photoproducts which are damaging to the DNA. The fourth option was topoisomerase DNA complex. It is again endogenous. It gets arise during the DNA replication and transcription when topoisomerases transiently cut the DNA to relieve supercoiling. So the function of the topoisomerase is to uncoil or unwind the DNA by nicking the DNA. Later on it gets sealed, but if it is not properly sealed, it can lead to the DNA damage. So again, this is also an endogenous source, not an exogenous source. So the correct option for this question is option C, ultraviolet radiation. I hope this question is clear. If you guys have any doubt, you can ask any question. If it is clear, we can move to our next question. Again, no question. So let's move to our next question. Question 2. Choose the correct sentence regarding DNA base mismatches. First option is DNA mismatches are defects in DNA that occur due to complementary base alignment in the same base pair of a DNA duplex. B. A transition is when a purine nucleotide is changed into a pyrimidine, or a pyrimidine nucleotide is changed into a purine. C option is transversion, or any mutation in which purine is replaced with a pyrimidine or vice versa. D option is transitions are more likely to change the amino acid sequence of protein than transversion. Please go through, take a while and go through the question and try to answer this question. Please try to answer this question. So we have one option, option C. Is there any other option that might be correct apart from option C? So let's look into each of the option. So the option 1, it states DNA mismatches are defect in a DNA that occurs due to complementary base alignment in the same base pair of a DNA duplex. Uh, it is a wrong statement because DNA mismatches, they are the defects in the DNA that occur due to non-complementary base, uh, which when incorrectly incorporated in the same base pair of a DNA duplex. So it should have been the non-complementary, but here it is written complementary base, so it is not a correct option. Then the option B, it states a transition is when a purine nucleotide is changed into a pyrimidine, or a pyrimidine nucleotide is changed into a purine. So transition is when a purine is changed into a purine, and pyrimidine, it gets changed into a pyrimidine. So it is again a wrong statement. While option C, it states transversion are any mutation in which purine is replaced with a pyrimidine or vice versa. So this option is correct. So transversion are any mutation in which a purine is replaced with a pyrimidine or vice versa. So D option is transition are more likely to change the amino acid sequence of a protein than the transversion. This option is also incorrect because in transition purine is replaced by a purine. Both purines are having the same structure, while the pyrimidine, they also have the same structure, but when purine gets replaced by pyrimidine, they are very highly different in structure. So the change in the amino acid are more prominent during the transversion than that of a transition. So the DNA mismatches, again, they are the defects in the DNA occurring due to non-complementary base alignment in the same base pair of a DNA duplex. So DNA mismatch they are different repairing mechanism, which we will look into later. What can be the repair mechanism? So here, as we have seen previously as well, here this G is replaced by A, so this is a DNA mismatch. So a transition is when a purine is changed into another purine. So the purines are A and G. As you can see, they have similar structure. They have two rings, while the pyrimidine, they have only one ring. So the pyrimidines have similar structure, while the purines have different same structure. So transition is when a purine is changed into another purine, when A is changed into G, or pyrimidine is changed into another pyrimidine, or C is replaced with T, or T is replaced with C. These are transition, and the transversion are any mutation in which a purine is replaced with a pyrimidine. A is either replaced with C, or A is either replaced with T, or it can be any other combination. And these transversion, they are more likely to change the amino acid sequence of the protein. So how these single base pair mutation, they leads to DNA damage. So let's look. So we have a wild type DNA sequence where, if you look here, we have G complementary to C. It is all fine. So then there is a single mutation occurred where this C is replaced via T. So this is a transition. I think again my screen sharing has been stopped. So this C is replaced by T. So while replication, we know that the double strand of the DNA gets separated and the new complementary strand is formed from one parental strand. So we now, after the incorporation of a different base, a wrong base pair, these strands would get separated, and here in place of G, the C would come, but now this strand has T. So while replication, the DNA polymerase will add A here. So it will result in a again the two strand, two DNA, which one copy of a DNA will have GC, the correct base pair match, while the other copy of a DNA will have a wrong base pair match, A and the T. So this is how a single base pair mismatch will lead to the mutation in the entire genome, will generate a mutant DNA. So this is how these single base pairs amplify itself while replication. So the correct option for this question is option C, transversion, or any mutation in which a purine is replaced with a pyrimidine or vice versa. I hope this question is clear. If you guys have any doubt in any question, you guys can interrupt in between any question. So I hope this question is clear. Okay, if it is clear, let's move to our next question. Question 3. Question 3 states, deamination is the process of removing dash from a molecule. A option is an amino group. B option is a carboxyl group. C is hydrogen. D is none of the above. Please try to answer this question. Okay, so we have one option, option A, an amino group. So this is kind of straightforward question. Can anyone give an example where if an amine group is replaced from a nucleotide, what is the change? Like any example one can give? What if we remove amine group from a cytosine? It will change into or turn into what nucleotide or a base? Or why we even focused? Why we even studying about the amination? What is the impact of deamination? So glutamate to alpha ketoglutarate is okay. So these are at the level of a protein or amino acid. So we are here talking about at the level of the DNA or a nucleotide. the correct option for this question is of course option A, an amino group. So let's look into how the deamination can lead to DNA damage. So again, the deamination is a process of removing an amino group from a molecule. So we know that in the structure of the nucleotide we have amine group. So this removal of this amine group from the nucleotide, it is the process of deamination. So when we remove an amine group from cytosine, it gets converted into uracil. And if we remove, if the amine group is removed from adenine, it gets converted into hypoxanthine. Or an amino group is removed for guanine, it turns into xanthine. Or if an amino group is removed from 5-methylcytosine, it turns into thiamine. And in the first lecture, we have studied about the genetic code and how the single mutation or like such deamination can lead to the incorporation of a different base or turning them into a different base pair, and it will lead to either truncation of a amino acid synthesis or it will lead to a different amino acid synthesis. So this is how a single or the removal of an amine group will change the protein sequence or will change the amino acid sequence. So this base deamination, it is more common than a single strand DNA in single stranded DNA than in a double stranded DNA, because in a single stranded DNA, so these are easier to be replaced because they are not they are not forming a bond with another base pair. So this C, okay, so here is this C is paired with G, so this is the cytosine and this is the guanine. So they have 3 bonds among hydrogen bond among these C and G. So after deamination, this C has turned to U, the uracil, and after replication, this G will be replaced by A, because as I have already told, during the replication the 2 strands will get separated and then the replication will occur. So the one DNA strand will have UA combination, the other the strand will have CG combination. So this is how a single base pair change or a single deamination will lead to the change in the DNA sequence. So the correct answer for this question is option A, an amino group. I hope this question is clear. Uracil is not found in DNA, it is generally found in RNA, but if we change, but these deamination will lead to the formation of uracil, so it will lead to the replication error as well. But these deamination can occur in RNA as well, so here it will directly lead to the change in protein sequence. So I hope this question is clear. Okay, if it is clear, let's move to our next question, question 4th. Question 4th states, which one of these is not a true regarding alkylating agents? Option A is so this question here we need to find not true regarding alkylating agents. The first option is alkylating agents transfer alkyl group to DNA. B option is simple methylating agents generate adducts at H and C atoms in DNA bases. C option is O alkylation are stronger mutagens where N alkylation are less mutagenic. D option is N alkylated purines are the primary product of DNA methylation. So please try to answer this question. Please try to answer this question. The answer can be wrong, it's fine, but please try. Okay, so let's look into each of the options. So, what? So, alkylating agents. What? So, alkylating agents they add alkyl groups such as methylethyl to the DNA base, altering their structure and potentially leading to the mutation. So, like we have seen, if the methyl group is added, this can lead to the change of one amino acid, one nucleotide to different nucleotide. So the alkylating agents can lead to the DNA damage, or it can lead to the mutation as well. So the B option was simple methylating agent generate adducts at H and C atoms in DNA bases. So these are incorrect option. So alkylation, it typically occurs at the electronegative atoms such as N oxygen. So these are the sites where this the alkylation occur. So atoms, they are not alkylated, and the C atoms alkylation is also very rare. So the common alkylation site is the N7O6 of guanine. So this is the N7, and this is okay. Again, sorry. So this is the N7, while this is the O6 of the guanine. So they are the common alkylation site, or the N3 of the adenine, etc. The third option was O alkylation are stronger mutagens, where N alkylation are less mutagenic. This all statement is also true. O alkylation, like O6 methyl guanine mispair during the replication, are highly mutagenic. N alkylation are more frequent but are often repaired and are more efficiently and often repaired quickly and more efficiently, and they are less mutagenic. D option is N alkylated purines are the primary product of DNA methylation. So this is also true. The most common methylation is the N7 and methyl guanine. So the correct option for this question, okay. So the alkylating agents, they can prevent the replication and the transcription. Once the base pair is alkylated, the cross bridge or the adducts are formed between the base pairs. So here you can see this G is cross linked with this G. So or there is the formation of the adducts which lead to the prevention of the replication process or the transcription process, or there might be leading to the mispairing, where here you can see this here you can see, so this C is getting changed into this T, so this base mismatch pair can also occur because of the alkylation of the base pair. So the correct option is option B. Simple methylating agents generate adducts at H and C atom in DNA bases, which is an incorrect statement. The correct position is N and O atoms. I hope this question is clear. Is there any doubt in the question, or we can move forward? Okay, let's move to our next question. Question 5. Question 5 states, name the process by which UV inactivated organisms restore their function by repairing UV induced DNA damages with the energy of near UV light. A is base excision repair, B is mismatch repair, C is translation synthesis, D is photoreactivation. Please try to answer this question. We have option D in the chat box. there any other option which might be correct apart from option D? So the correct option for this question is option D only, photoreactivation. So the correct option for this question is option D, photoreactivation. So let's look into the concept. So like we have already seen that there are certain endogenous as well exogenous factor which leads to the DNA damaging via either the single base pair mismatch or the single strand break or the double strand of the DNA break or the cross linking between the base pair between the nucleotides or the formation of the adducts. So there are certain cellular machinery in an organism which can repair these DNA damage or these mismatches or single stranded breakage. So there are different type of DNA breakage repair, photoreactivation, the base excision repair which repair the single strand break, or the mismatch repair which mismatch mediated repair which repair the base mismatch DNA damage, or the homologous recombination or the non homologous recombination which repairs the double stranded DNA break, or the non homologous enjoining which repairs the cross linking among or between the nucleotides, or the nucleotide excision repair which repair the adduct the DNA damaged by the adduct formation. So there are different sort of different type of DNA breakage repair mechanisms. So the photoreactivation, it is the mechanism which directly repairs the UV induced DNA damage using the visible or near UV light, and with the help of an enzyme which is called DNA photolyse. So when the UV the first when after the exposure of UV light, there is uh, the formation of the cyclobutane pyrimidine dimer. So these dimer distort the DNA helix, and it leads to the blocking of the replication and the transcription. So this enzyme, the photolyase enzyme, it detects these cyclobutane pyrimid dimers and goes scans and binds specifically to the dimerized bases. And this enzyme, it has the two cofactor. The one is FADH minus, it is essential for the catalysis, and one is MTHF. It absorbs the photon and transfer energy to FADH minus. So the process happens here when this enzyme is bound to the DNA and is exposed to the visible or the near UV light. This light harvesting cofactor, it absorbs a photon and the energy is transferred to FAD minus. So this excited FAD minus, it donates the electrons to the dimer, and this breaks the abnormal cyclobutane ring. So here you can see this ring is getting break when this FETH minus, it donates an electron to the dimer. So after the dimer, after this the dimer is repaired and the electron is returned to FETH minus, resetting the enzyme, and this uh, the enzyme is also released, and the photolyase, it dissociates from the DNA. So this is how this photolyase, it detects the dimer formation, and it leads to the breakage of this dimer formation, and again restores the normal base pairing. So the correct option for this question is this photoreactivation process. It corrects the UV. It corrects the UV induced DNA breakage with the help of the near UV light. So I hope this question is clear. I think people are still joining in. So those who are joining in late, please try to join early. So it's already been 1 hour of the class. So I hope this question is clear. So let's move to our next question. Question 6. I think my screen sharing stopped. So the next question is question 6. Name the enzyme involved in base excision repair. First option is DNA glycosylase. B option is DNA polymerase. C option is DNA topoisomerase. D option is the RNA polymerase. Please try to answer this question. We have one option in the chat box, option A. Is there any other option that might be correct apart from option A? The correct option for this question is option A only, DNA glycosylase. So this base excision repair, it is a DNA repair mechanism that corrects small non-helix distorting base lesions such as deaminated bases, oxidized bases, alkylated bases, or AP sites, apurinic or apyrimidinic, where there is absent of either purine or pyrimidine base in the DNA. So there are two type of base excision repair. One is a short patch repair, one is a long patch repair. Short patch is where only a single nucleotide change is there, while the long patch is there certain region is the long patch repair. Okay, let's see the first see the difference so you guys will know. So in a short patch base pair excision repair, the first DNA damage site is recognized by the enzyme DNA glycosylase. It is a specific glycosylase recognize and remove a damaged base, creating a AP site or a purinic or a pyrimidinic site. So now the base pair, the base is removed from this site. Now there is no base is present. Then the second step is backbone cleavage, where AP endonuclease it cleaves the phosphodiester bond, the 5 prime, to the AP site, generating a single strand break with 3 prime OH and 5 prime deoxyribose phosphate. Now the nucleotide insertion. Now we have a DNA polymerase beta. It removes the 5 prime deoxyribose phosphate group and insert a correct base using the 3 prime OH as a primer. So now this DNA ligase 3 with XRCC1 seals the nick in the DNA backbone and it is completing the repair. So this is the short patch repair. In case of a long patch repair, till backbone cleavage, it is the same process, but during the nucleotide insertion, so sometimes it happens is okay. My screen is okay. So sometimes what happens is this 5 prime deoxyribose phosphate group is resistant to the DNA polymerase beta because it might be oxidized or reduced. So here the lesions or the breakage is more complex, and this short patch DNA repair it fails to repair. So here again the damage recognition is same via the glycosylase. Then the backbone cleavage is done by AP endonuclease. Then the strand displacement synthesis, where now we have a DNA polymerase sigma or eta. So they extend the DNA strand by inserting 2 to 10 nucleotide and displacing this old strand. Now this flap removal occur with the help of FEN and PCNA, and now again the ligase is used to seal the breakage. Then now we have again the repaired DNA strand. So this is how the short patch and the long patch base excision repair occur. So only the differences in the DNA polymerase, which DNA polymerase is used. In short patch repair we have DNA polymerase beta, while in the long patch repair we have DNA polymerase sigma or eta. So the correct option for this is name the enzyme involved in the base excision repair. So DNA glycosylase, it recognizes the DNA damage site. B option is DNA polymerase, but it is not specific what DNA polymerase is there, so we will go with the option DNA glycosylase. So DNA topoisomerase and RNA polymerase, they are not involved in the base excision repair, they are involved in the DNA replication. Topoisomerase is involved in DNA replication via unwinding of the DNA, while the RNA polymerase is involved in the transcription. So correct option is DNA glycosylase. So I hope this question is clear. Yes, DNA polymerase also helps in proofreading. Because it has 3 prime to 5 prime exonuclease activity, so it also helps in proofreading. Also depending on which DNA polymerase. Okay, so if it is clear, let's move to our next question, question 7. Question 7 states, which of these is not involved in single strand break repair? Please go through the question and try to answer this question. We have one option, option A. Is there any other option that might be correct apart from option A? So we have another option, option B. We have another option in chat box, option C. Okay, so the correct option for this question is option C. Uh, TLS polymerase is the correct answer. It is not involved in single strand break repair. So let's look into the pathway for this repair pathway. So the single strand break, it is a discontinuity in one strand of a DNA double helix. It is a common DNA damage. These are caused by either oxidative stress, ionizing radiation, topoisomerase malfunction, or base excision repair intermediates. So there are they can occur by the oxygen species or the oxidative stress or the topoisomerase 1. So there are two different uh, uh, mechanism to correct these single strand break. So the one is occurred by the reactive oxygen species. So here this PRP1 or poly ADP ribose polymerase 1, it detects and bind to the single strand break and synthesize poly ADP ribose chain or pyrylation using NAD+. These part chain, it recruits an organized repair factor to the break site. So then this SSBs, they have the non-ligatable DNA ends, the 3 phosphate as well as the 3 prime phosphate as well as the 5 prime hydroxyl group, they are absent. So now we have the enzyme, the polynucleotide kinase 3 phosphatase. It converts the 3 prime phosphate to 3 prime hydroxyl as well as 5 prime hydroxyl to 5 prime phosphate to make the end suitable for DNA synthesis and ligation. Then this gap filling occur with the help of XRCC. These are X-ray repair cross complementing protein 1. They act as the scaffold protein. They are they interact with DNA polymerase beta as well as the ligase to seal the breakage or to ligate the strand. The ligation occur between the 3 prime OH as well as the 5 prime phosphate group. Then the DNA ligase 3 is seals the gap or makes the bond between these two. While the single strand break caused by the top 1 activity, so here the source is topoisomerase 1. It relieves the DNA supercoiling by creating transient single strand break. Normally what happens is this top 1 religates the break after relaxation, but abortive top 1 activity either due to the collision with the replication fork or the drug inhibition, it leaves the covalent top 1 and the DNA complex. So this is not being removed, so they have formed a complex top 1 and a DNA. Then further it will lead to the hindrance in the replication process as well as transcription. So these sort of single strand break, the single strand break with this top 1 cleavage complex, they are covalently attached to the 3 prime end, the 3 prime phosphotyrosyl bond, blocking the further processing. I think my screen is. Then again we have this PRP1. It again recognizing the break. It also recognizes the break in case of reactive oxygen species single stranded breakage. So this again sends the break in case of the top 1 isomerase attachment. Then it removes the then the removal of the top 1 adduct with the help of this TDP1. It hydrolyzes the 3 prime phosphotyrosyl bond, releasing the top 1 fragment and leaving the 3 prime phosphate. So then end processing occur with the help of PNKP. It converts the 3 prime to 3 prime OH. So now we have 3 prime OH and the 5 prime phosphate. This PNPK, it converts the 3 prime phosphate to 3 prime OH as well as the 5 prime OH to 5 prime phosphate. So it is not written here. So this gap filling, further this gap filling will occur with the help of the XRCC1 and the DNA polymerase, which fills the gap as further this nick is sealed with the help of the ligase 3. So this is how this single strand DNA breaks are repaired with these protein complexes. So the basic principle of each of the repair is, first it has to be recognized by certain protein. What type of there are different protein which are involved in the recognition of different DNA damage. So now then we have to uh, fill the DNA damage site with the help of either DNA polymerase, and then we have to have the 3' OH as well as 5' phosphate, where the DNA ligase come and it ligate the DNA base pairs. So this is the basic principle. So there are different proteins which are involved in the different mechanism. So you can try to remember these protein. So in this case, the TLS polymerase is not involved. It functions in the translation DNA synthesis, a damage tolerance mechanism during replication that allow the DNA polymerase to bypass the bulky lesions such as thymine dimer. So it is not involved in the SSB repair pathway. So the correct option for this question is TLS polymerase. So I hope this question is clear. Okay, so let's move to our next question. Question 8 states, which of these disorders is associated with severe photosensitivity resulting in sunburn and skin pigmentary changes? Please go through the option and try to answer this question. We have option B in the chat box. Is there any other option that might be correct apart from option B? Okay, so the correct option for this question is option B, xeroderma pigmentosum. So the key feature for this is severe photosensitivity. The individual experience extreme sunburn, freckling, skin pigmentary changes, uh, high risk of skin cancer even after minimal sun exposure. How it is caused? It is an inherited defects in gene involved in a nucleotide excision repair. It is especially in the repair of UV induced DNA lesions like thymine dimer. So this uh, this occur via there is a defect in a gene which involve in a repair pathway, which is nucleotide excision repair pathway. It helps in the breakage of the thymine dimer formation, but if it is defected, able to it is not able to correct these defects. So the effect is the inability to repair DNA damage caused by UV radiation, leading to mutation and early skin aging and cancer. So the other are not the correct answer. So the correct answer for this question is option B, xeroderma pigmentosa. I am not sure why people are still joining in. So let's move to our next question. Question 9. Choose the correct group of protein involved in E. coli DNA mismatch repair pathway. Please take a few minutes and go through the question and try to answer this question as well. We have one option A. So we have option A in the chat box. Is there any other option that might be correct apart from option A? Okay, so the correct option is option A. So let's look into the concept for mismatch repair pathway in E. coli. So the mismatch repair, it is to correct the base mismatches or a small insertion deletion loop that occur during the DNA replication and recombination. So in E. coli, the E. coli distinguishes the newly synthesis strand which might contain the error by the fact that it is unmethylated, while the template strand is methylated at GATC site. So why these methylation occur in the E. coli genome? It is because this E. coli has the immune mechanism against the viruses which infects these bacteria. So they have certain enzymes which are restriction endonucleases enzymes which cuts the incoming DNA from the virus which infects them. So what they do is they methylate their own DNA so that these E. coli restriction enzyme, they won't cut their own DNA. So they methylate their DNA. So, but the virus DNA that will be infecting the E. coli won't be having the methylated DNA. So they recognize the DNA and cuts so that the replication of the virus it is not completed inside the E. coli or the inside the bacteria. So that is why they methylate their own DNA. So to prevent themselves from the restriction endonucleases. So so these E. coli distinguishes so these DNA gets methylated after the replication. So just after the completion of the replication, the new strand is not methylated. So this is how they distinguishes between the newly strand DNA and the template strand. So and if they can see any mismatch in the newly strand DNA, so there is a mechanism which is the mismatch repair mechanism. So where these mismatches are recognized by the mute-S enzyme, it scans the DNA and recognizes the mismatch base pair. It binds tightly to the mismatch site. Then it recruits the mute L and mute H. The mute L, it binds to the mute S. It forms the mute L and the mute S complex. This complex activates the mute H. It is a latent endonuclease. So the nucleases are the enzyme which cuts the DNA. So the strand discrimination occur where the mute H, it discriminates the newly synthesized strand by identifying the unmethylated GATC sequence. A parental strand is methylated by an enzyme dimethylase. This mute H then nicks the unmethylated daughter strand at the nearest hemimethylated GAT site. Then the DNA unwinding occur. From the nick, this DNA helicase 2 or UVRD, it unwinds the DNA towards the mismatch. Then excision of the error segment. This unbound strand containing the mismatch is removed by the enzyme exonuclease, exo1, exo6, reg-J or exo-X, depending on the direction of excision, either from the 5' to 3' or 3' to 5'. Then again the DNA resynthesis occur. The resulting gap is filled by the DNA polymerase 3 holoenzyme using the parental strand as a template, and the next healing is done by the DNA ligase. So the correct sequence is the mute S and then the mute L, mute H, then the DNA helicase 2, exonuclease, DNA polymerase 3 holoenzyme, and DNA ligase. So the option 1 is correct. So I hope this question is clear. I hope the question is clear. So let's move to the next question, question 10. Uh, Question 10 states which one of these is the enzyme involved in repairing DNA damage caused by exposure of UV light. A is DNA glycosylase, B is DNA helicase, D is DNA ligase, C is DNA ligase, D is DNA photolase. Please try to answer this question. We have one option D in the chat box. So we have already seen this question. So the correct option for this question is option D, DNA photolase. So as we have already discussed, this DNA damage leads to formation of the cyclobutane pyrimidine dimer, and this enzyme it recognizes these dimer and also helps in breaking of these DNA dimer. So we have already seen the mechanism of this in the previous question. So the correct option for this question is option D, DNA photolase. So while DNA glycosylase, it was involved in base excision repair. It was involved in the recognition of DNA damage in base excision repair. DNA helicase, it unwinds the DNA during the replication or repair, while the DNA ligase, we have seen it seals nicks in DNA after repair or replication. DNA photolase, it is directly repair the UV induced pyrimidine dimer. So the correct option for this question is option D, DNA photolase. I hope this question is clear. So if it is clear, so this was our last question for today's session. So if you guys have any doubt in any of the question, you guys can ask. So if you guys don't have any questions, so we will wait for our problem solving session third in the next week at the same time on Tuesday. So if you guys have any doubt, you guys can ask. If you guys don't have any doubt, thank you for joining in. You can leave the meeting.